Hello, everybody. I am Mercedes Gomez. I am postdoc in the Laboratory for Evolutionary and Ecological Immunology with the PI Michal Vinkler. I will introduce the result for analysis of cytokine expression profiling after induced inflammation in non-poultry avian model. What cytokines are? Cytokines play a key role in the inflammatory process that is defined by the balance in inflammatory regulation. When the balance is not correct, it leads to a disease or disorder. This regulation can be then targeted in therapy for chronic inflammatory diseases or uncontrolled inflammatory episodes caused by zoonotic infections. Cytokines are proteins synthesized by immunological cells, and at the same time, they are responsible for immune cells to be differentiated, proliferated, or secrete one type of antibody or another. They are crucial components to initiate and regulate immune response, both innate and adaptive. It's so that this molecule travels around the body. Oops. We are interested we are focusing pro-inflammatory cytokines. For example, interleukin-1 beta is macrophage cytokine, and this is primarily associated with acute and chronic inflammation. Also, it's involved in a variety of cellular activity, including cell proliferation, differentiation, and apoptosis. Other pro-inflammatory cytokine is interleukin-6. This cytokine is involved in the maturation of B cells. CXCL1 and CXCL2 are two orthologous genes for interleukin-8 in birth. These chemokines are chemoattractant activators of heterophiles in birth and monocytes. The aim is to describe the parent peripheral and systemic inflammation across tissues and validates protocol for estimate of cytokines expression change indicating different inflammation roles. In our study, we use non-poultry avian model like Badger. The genome of this parrot has been sequenced and annotated. A number of studies providing insight into the behavioral similarities between human and parrot to understand mechanisms of the evolution of cognitive abilities as well as understanding illness without a cure. We use 30 individuals. We inject intra-abdominally LPS to induce systemic stimulation. And subcutaneously, we inject the LPS to induce peripheral stimulation to study a local immunological response. Also, we introducing our experiment different time point scale for euthanization at 3 hours, 6 hours, 12 hours, 24 hours, and 48 hours. After induced inflammation, we are interested to study brain, ileum, large intestine, and skin, and the connection between them. For this, we perform real-time PCR. We measure the RNA expression for interleukin-1 beta, interleukin-6, interleukin-18, CXCL1, and CXCL2. The principal component analysis tell us that the pro-inflammatory immunological genes by their expression are grouped in 3 main clusters. Also, we can see that this cluster follow the same pattern in the different tissues, like skin, brain, large intestine, and ileum. As you can see in the figure, all the pro-inflammatory cytokines genes show a statistically significant positive correlation. Keeping this result in mind, we studied deeply 2 genes from different PCR clusters, like interleukin-1 beta and interleukin-6. When we want to see the result to respond for systemic immunological response, we can see in the panel how the pro-inflammatory cytokines, interleukin-1 beta, show different level of expression between control and treatment birth across all the tissues, skin, brain, large intestine, and ileum. But there is no significant difference for different point times. Also, the gene upregulation was depending on the tissue. The expression of interleukin-1 beta was higher in skin, followed by ileum, large intestine, and brain. In contrast, what is happening for interleukin-1 beta, interleukin-6 changed the expression over the time. The figure shows how in treatment birth, the peak expression was observed at 3 hours after LPS stimulation and decreased at 24 hours and 48 hours. While in control birth, the interleukin-6 gene expression is stable over the time. As with interleukin-1 beta, interleukin-6 finds a statistically significant difference in expression among tissues. Now I will explain what is happening with local immunological response when we compare control wings and treatment wings. In the figure, we show in red the expression for treatment wing and in blue for control wing. Also, we use triangle for stimulated birth and single and circle for control birth. Interleukin-1 beta is upregulated in treatment wing for stimulated birth. Also, control wing for this parrot is upregulated with respect to the same wing in control birth. The tendency of interleukin-6 gene expression in skin was dependent on birth treatment. As the left figure shows, interleukin-6 is upregulated in treatment wing for both stimulated and controlled birth. Our conclusions are that pro-inflammatory cytokines genes follow the same regulation pattern in brain, intestine, and skin. These genes are strongly correlated with each other. Interleukin-1 beta and interleukin-6 are involved in inflammatory, and they are marker candidates in brain and intestine. The upregulated peak is at 3 hours after stimulation. The peripheral immune response is upregulated by systemic stimulation. Just say thanks to the evolutionary and ecological immunology group in Charles University, and thank you everybody for attending.